The overall goal of this procedure is to minimally invasively inject a drug-loaded supramolecular hydrogel into an infarcted pig heart using a long, flexible catheter. This is accomplished by first preparing a formulation of hydrogelator and the desired drug. The second step is to investigate the properties of the hydrogel and the rate of drug release in an in vitro setting. Then, 4 weeks after myocardial infarction, an electromechanical map of the pig's left ventricle is made using data points taken from the endocardial surface. Then, the so-called hibernating myocardium is targeted for local injections with drug-loaded hydrogel. This convenient formulation and application procedure can lead to uncovering new therapies for myocardial infarction and so forth.
The main advantage of this technique over existing percutaneous approaches like intracoronary fusion are 1. The catheter application of the hydrogel, and 2. The possibility of treatment planning, which allows us to guide the catheter to the area of interest.
This method can help answer key questions in the study and treatment of myocardial infarction, such as potential advantages of injecting a mechanically supporting material and the effectiveness of a sustained local drug delivery.
The implications of this technique extend toward therapy in myocardial infarction, because the electromechanical mapping allows for identification of the infarcted region, and subsequent guided site-specific injection of hydrogel allows for local drug delivery.
Generally, individuals new to this method will struggle because this technique requires practical skills with subtle movements of the catheter while monitoring the stability parameters.
Visual demonstration of this method is critical, as both the formulation of drug-loaded hydrogels and the intermyocardial injection via a catheter